Afternoon, everyone. I am your host, Shailesh Shukla. We're starting the webinar very shortly. Thank you for joining us today. Afternoon, everyone. I hope you're doing well. On behalf of Advent Informatics, I warmly welcome you to today's insightful webinar on a focused and highly relevant topic, which is decoding real life in real time, the spike protein and ACE2 interaction. We like to have you with us today, and we look forward to a session full of practical demonstration, expert tips, and interactive learning. Before we begin today's session, allow me to briefly introduce the organization behind this event, Advent Informatics Private Limited, headquartered in Pune. Advent Informatics is a leading distributor of advanced life science software solutions. Our core expertise spans across chemoinformatics, molecular modeling, medicinal chemistry, chemical data curation, and biological information management. We serve a wide range of clients, including leading pharmaceutical and chemical companies, direct research organizations, academic institutions, and government R&D labs across India. Our mission is to bridge the gap between complex scientific challenges and advanced software solutions by, enabling researchers the world-class technologies for efficient discovery and innovation. Proudly represent globally recognized technologies partners such as Molsoft, Chemaxon, Elsevier, Campus, Certara, Transgene IT Solution, and Keymotive, helping drive innovation across the life science and drug discovery domains. Now, my pleasure to introduce our speaker for today, Mr. Nachiket Joshi, Junior Application Scientist at Advent Informatics Private Limited. Mr. Nachiket Joshi is currently pursuing his PhD in pharmaceutical science from SVKM, NIMS, and MIMS, with a strong academic background in pharmaceutical chemistry and hands-on expertise in molecular modeling. Specialized in techniques such as molecular modeling, molecular dynamics, QSAR, pharmacophore-based screening, and virtual ligand screening. The perfect guide for today's session on decoding real life in real time, the spike protein and ACE2 interaction. For this session, if you have any queries, please feel free to type them in the chat box. Join me in warmly welcoming Mr. Nachiket Joshi. Nachiket, the floor is yours.
Thanks, Shailesh. That was a great introduction. Thank you so much. So, hello everyone. Welcome to today's webinar titled Decoding Real Life in Real Time, the Spike Protein and ACE2 Interaction. I'm your friend, Mr. Nachiket Joshi, working as Junior Application Scientist at Advent Informatics. Yeah. So Advent Informatics is a company which was founded in 2016. We are based in Pune, and we are prominent distributors of all these softwares, as Shailesh has already mentioned. So Molsoft is a molecular modeling sort of a tool. Then we have Chemaxon, which is like, if you know Marvin Sketch, which is like a chemistry drawing software. That is from Chemaxon. We have SCM, which is quantum chemistry based solutions and computational fluid dynamics based solutions. Then we have an AI based forward reaction predictor, which is like ChemPass. We have data analysis with Certara, laboratory information management system with Transgene, and recently we have partnered with Elsevier, which has 4 different databases: Reaxys, Pharmapendium, Embase, and Embiology. So broadly covering a big range of life science databases there. As well as you can also do retrosynthesis, AI-based retrosynthesis using Elsevier's Reaxys. So we are highly appreciated for our support by our clients. And when I say that, in 2024, when we carried out a survey, so our users rated us 5 out of 5 stars for our responsiveness. So we respond to all of your queries within a day itself. We provide technical support for all of the softwares that we have mentioned. So a bit more about Molsoft. So Molsoft is a 32-year-old company. Its code was written even before Autodoc's first release. The early adapters were BMS and Novartis. And we have consistently outperformed commercial or open source softwares, especially in docking, accuracy, post predictions, RMSD, and binding affinity. So this is a white paper which was published by AstraZeneca, and it is a comparative study between ICM and other softwares. So let me just let me just focus on that. Yeah. So if you see in this particular image, you can see about the cutoff of 2 angstrom for RMSD. You can see there are a lot of different confirmations, many more confirmations compared to ICM for other softwares. Whereas with ICM, you can see only a small number of confirmations which are above that RMSD cutoff range. So if you don't know about MoleSoft, you can always do a Google search. You will find about 6,500 plus citations currently, and there are about 200 plus hits in Nature journals, about 1,300 plus hits in Science Direct, and all such well-known publishers that we commonly know now. Yeah. So this is a global subset of most of users. We have industries like Pfizer, Novartis, Eli Lilly, Merck, AstraZeneca, which are using MoleSoft. Along with them, very reputed academic institutions like NIH, National Cancer Institute, Stanford University, University of Toronto, and you can see multiple other users here. So coming to India, we have industries which are driving R&D in India, like Syngene, Aragen, Zydus, Origin Oncology, Origin Think Molecular, Jubilant Biosciences, and PL Health Sciences. So all of them, they are using MoleSoft. Other than that, we also have academic institutions like Naipur Mohali, Naipur Guwahati, IIT Gandhinagar, IIT Dharwad, Central University of Punjab, Chanakya University, GSFC University, Geetam University. All of them, they are also using MoleSoft, and this is just a small subset of users. The list is even bigger. So as I said, if you don't know about MoleSoft, you can always do a search and you will find articles in very reputed journals like Blood, Nature, Scientific Reports. So if you want to know more about these publications, you can use this link to access our publications. So if you're wondering how this work was done, the protein docking, or the work that we have undertook, it was done using these MoleSoft modules. So there are two basic modules that we have. The basic module is ICM Pro plus homology modeling. Then we have an ICM Pro plus homology modeling plus VLH, which we call it as our full package. And independently, if you want to carry out chem informatics based work, you can also use the standalone tools, which are ICM Chemist and ICM Chemist Pro. So in ICM Pro, you can do multiple different types of docking, like small molecule docking, protein docking, which has been done in this particular case study, induced fit docking, peptide docking. So you can see there are multiple different things that you can do with one particular tool itself, which is ICM Pro. We can also do MD simulations, homology modeling, PROTAC modeling. Broad range of tools are there. You also have ICM VLS. So if you go for the full package, you can go ahead with all different types of virtual ligand screening. Then you can do PCA analysis, 2D, 3D QSAR, SAR based analysis as well, pharmacophore searches as well, AIML based admit predictors. You can use that as well. So all of these tools, they are available with us. So coming to today's topic, which is protein docking and exploring our specific interaction between the spike protein RBD, that is the receptor binding domain, and the ACE2 protein, which is present in the humans. So before that, I just want to give you a short landscape about ICM MoleSoft's protein docking module. So ICM MoleSoft, as I said, it is a 32-year-old company. So we essentially work with internal coordinates. So internal coordinates are composed of torsional angles, dihedral angles, phase dihedral angles, so all of those. So what happens is, since we do not use these Cartesian coordinates, which all the other softwares they use, so this saves a lot of time and it does not compromise on the accuracy. So this was started in 1994. Then we have participated in certain challenges like the blind docking challenge, Capri challenge as well, wherein we have been the top performers. Additionally, we have also added tools like ICM Disco, which is like a sidechain optimization tool, and ODAs, which is optimal docking areas tool. So I'm going to talk about ODAs a little bit more in the coming slides. So basically, what are protein interactions? So protein interactions, they are specific physical associations between two or more proteins that enable our body to carry out the perform our biological functions together. So they are very important in carrying out our metabolic functions or biological functions which are needed for our body. But if these protein-protein interactions they go hayway, that is, if they have if there are abnormal protein-protein interactions, they are linked to a wide range of diseases like cancer, neurodegenerative diseases, autoimmune disease, or viral infection, like we're going to see in today's session. They so these type of interactions, the protein-protein interactions, they are mediated through different molecular forces, primarily by hydrophobic interactions, electrostatic interactions, but also by hydrogen bonding, van der Waals interactions, as well as salt bridge interactions. So what are the reasons now to perform this protein-protein docking? So protein-protein docking is usually performed because there is unavailability of the crystal structures for protein-protein complexes. So to have two flexible moieties, which are proteins, with each other is extremely difficult. And that is why, to understand these interactions, you need to carry out protein-protein docking, which is a much simpler approach compared to any wet lab sort of a study. So to understand interactions of these two proteins with each other, two or more proteins with each other, you need to carry out this protein-protein docking. And as I said, the inherent flexibility of these protein structures in the solution phase, this makes it very difficult for you to avail these crystal structures, and that is why you need protein-protein docking and then further analysis of those interactions. So talking about the SARS-CoV-2 ACE binding interaction. So what happens is, this is the mechanism of viral entry for SARS-CoV-2. So if you see in this particular diagram, so very elaborate diagram by the way, and if you see you can see the ACE receptor here, ACE2 receptor. This is present in our body, and you see how the virus is enabling the interaction through this ACE2 receptor and the spike protein RBD, that is the receptor binding domain. And if you see how it is progressing, initially the interaction is through this ACE2 receptor and the spike protein, and after that the entire virus materials, they are ingested into the host, and then it is again went ahead for infection or transfection, all of those, and then you get that SARS-CoV-2 disease. So if you see here clearly, it is showing that the ACE2 and spike protein interaction, this is very critical for the entry of the virus into the host cell. So if you can design something which design drugs which can stop this particular interaction, then we can have like an anti-COVID sort of a drug. So that can be a drug design strategy if you want to take that ahead. So coming to the case study for today, so ACE2 spike RBD protein, it is a very well-known protein-protein interaction. As I told, it is essential for viral entry into the host. So what have we done is, we have taken individual proteins of spike protein RBD as well as ACE2 receptor, and then we have docked these two proteins to form a particular complex. So after the docking results, we have selected the best possible complex based on the total energy value, that is with the least energy. It was selected for further studies, and then the stability of that best complex, it was analyzed by running 100 nanosecond MD simulation across 2000 trajectory frames. So this was the overall protein-protein docking workflow, and the further MD analysis also that we took ahead. So firstly, we imported the protein structures. So the individual structures with these PDB IDs, 1R42 is the PDB ID for ACE2 protein, and 7L4Z is the PDB ID that we have undertook for SARS-CoV-2 spike protein RBD. After that, we calculated the ODAs, which is optimal docking areas. So since protein-protein interactions, they do not happen into the deeper surfaces of the protein, they are very surface-based interactions. You need to carry out a prediction, or you need to know which are the hotspot residues present on these protein structures. So to calculate these hotspot residues, what we have in ICM MolSoft is the ODAs calculation, that is the optimal docking areas. So it will give a bunch of residues which can have these interactions, or which are more probable to form interactions in both these proteins. And then a protein-protein docking protocol was carried out using the information of these ODAs that we observed. Furthermore, docking validation was carried out for the dock complex against 6M0J PDB ID, which is essentially a PDB ID which already has like it is already deposited with the structure or the complex of SARS-CoV-2 and ACE2 protein. And then we went ahead with MD simulations. So this is the entire protocol that we have followed. So let me take you to my software and let us see it visually now. Yeah. So if I double click on the desktop icon for ICM Pro, I will find this sort of window. The top part of this window is the header section. This is my workspace panel. This is my graphical window, and here at the bottom is my terminal. So terminal is like the command line version for the software. So the workspace panel and graphical window, they are very interactive. So anything that you click in the workspace panel will be displayed in the graphical window and vice versa. So the best thing about the software is that there are multiple different databases that are included or incorporated into the software. So you do not need to go anywhere else in order to access anything. So that makes it very easy for you to just search your PDB here. So right now I'm just going to search my PDB, that is 1R42, and click on enter. So this is my crystal structure, X-ray crystal structure. So there are multiple different fragments, and I'm just going to consider the A chain. So I'm just going to go ahead and delete all these. So you can just give a right click and click on delete. So now, since I told that we do not work with Cartesian coordinates, we work with internal coordinates. The first step is to convert this XR crystal structure into an ICM object. So to do that, what we can do is just give a right click and click on convert PDB. So after you click on convert PDB, you can see a broad range of options here. So you can opt to keep tight waters. So tight water molecules are those water molecules which form three or more hydrogen bonding interactions in the protein. You can delete all water molecules. You can delete loose water molecules. So loose water molecules are those which do not form more than one hydrogen bonding interaction in the protein structure. Or you can also keep all water molecules depending upon your needs. So I am just going to keep it at default, which is at keep tight waters. Then the next option is to optimize hydrogen. So since hydrogen atoms are very, very small, in the X-ray crystal structure, they are not resolved. So the software now will optimize these hydrogens. It will add the hydrogen atoms and it will optimize the geometry and conformation for these hydrogen atoms. Then you can also optimize these 5 amino acid residues. So these 5 amino acid residues, they are present in different tautomeric forms. So you need to consider all of these tautomeric forms for an accurate result. So you can click on this option as well. Then this is a very straightforward option wherein you can replace the original. So the XR crystal structure will be replaced by the ICM object. Display the result. This option is also going to be ticked. Hide side chain residues, like missing side chains. So if you have some missing side chains and if you want to hide them in the graphical view, you can use this particular option. You can also assign secondary structures. So you can, when we are converting this XR structure into an ICM object, the software will by itself assign which amino acid residues are involved in the helix, which are involved in the beta pleated sheets, or which are involved in the loops. So all of that will be done using this particular option. You can also predict formal charges in the ligand and also carry out optimized ligand tautomer. So I'm just going to click on OK now. So now you can see there are some calculations or changes that are going on. You can see that in the terminal. Yeah. So my structure has now been converted. The XR structure has been converted into an ICM object now. So in the same way, I'm just going to load my other protein, which is 7L4Z, and press an enter. Now you can see there are multiple different chains here as well. So I'm just going to need the B chain. So let me go ahead and delete all the other ones. Yeah. Yeah. So again, now what I'll do is I'll convert this particular XR structure into an ICM object again using the same options that we had already seen. Just click on OK. So this is my spike protein RBD structure, and This is my ACE2 protein structure. So now if you see, since this SARS-CoV protein structure is a smaller structure, this will act like a ligand and the ACE2 will act like a protein. So now what we will do is we'll go ahead and predict the ODAs, which is the optimal docking areas. So if you, to do that, what we'll do is just go to tools, we'll select this particular SARS-CoV-2 structure, go to tools, 3D predict, protein interface by ODAs, selection by default, and I'll use these options and I'll just click on OK. So now it is going to calculate me ODAs. Yeah. So for all the residues, it will give an average ODA sort of a value. So let me show you that. Yeah. So you can see reddish colored balls and blue colored balls as well. So these reddish colored balls, they are essentially the more important residues which can be involved in potential binding interactions. So you need to consider these residues. So based on that, now if you filter this, if you give a right click, it will filter it in on the basis of average ODAs. So the higher the value of average ODA, the more probable is its involvement in forming binding interactions. So if you see, there are these residues which are having a higher probability of forming bonding interactions. So now what we'll do is we'll just label this mesh as specifically for 7L4Z. And we'll just carry out the same procedure that we carried out with the other protein. So go to tools, 3D predict protein interface by ODA, and I'm just going to click on OK. Yeah. Again, if you can arrange it in terms of average ODA, let me show you that. So this is your ACE2 structure, and this is the ODA for this structure. You can see some probable residues here as well. Yeah, these are these residues that are there in the red colored balls. So now with the information of these residues, what I'm going to do is I'm just going to set up my protein-protein docking protocol. So to do that, I'll go to docking. I'll click on protein-protein and I'll click on this option, which is run global FFT doc. So as I said, my receptor object is the ACE2 protein. My ligand object is my SARS-CoV-2 spike RBD protein. So I'll select those particular PDBs here. Now for sampling precision, you have four different options. You can go ahead with a quick test mode, a coarse method, a regular method, or a fine method. So based on the method that you select, the sampling precision will be higher. So if you select a fine sort of a sampling precision method, the number of confirmations that will be generated will be a lot more. So it will explore the entirety of the conformational space. Other than that, we'll use an object for evaluation here as 6M0J. So this is your complex which is already loaded in the PDB database, which has both the structures of the SARS-CoV protein as well as the ACE2 protein. So here now again, if you want to select different residues for your binding, there are two different options. So if you do not have an idea about the binding site residues, then you can straight away go with a global sort of a selection, which will scan the entire surfaces of both these proteins. Or you can also choose the residues that we have selected using ODAs. So to do that, what we have to do is first go into the ligand, that is our SARS-CoV-2 protein. So I'll just give a click here, right click, and click on neighbors. Sorry. Yeah. So go to select by residue numbers. Now I can select these top 5 or 6 residue numbers depending upon my need. So what I'm just going to do is write these residue numbers here: 489, 484, 486, 487, 493. So I've selected these top 5 residues and click on OK. Now you need to select these residues in orange selection, if you see here, for your ligand, and green selection for your receptor. So now to convert this selection into an orange selection, you have to use this particular option, which allows us to make multiple different selections at the same time. If I click on this, now it has converted into orange selection. Now I can go ahead and select this. So the ligand residues now I have selected as per the ODAs. Now I'll select my receptor residues. So for that, I'll go into this particular table, and again, I'll do the same thing. Select residue numbers. Again, I'll just write down the residue numbers that I am seeing on the screen. These ones: 504, 350, 273. And if I click on OK, now it will automatically be selected in green color notation, if you see here. So now I can just run my protein docking. If I want to increase the number of cores, I can also use these core options that I have. I can run the protein docking up to 8 cores. So after this, I can just click on OK. But I'm not going to run this protocol because it can take up some amount of time. I'm going to take you through the results that are obtained. Yeah. So these are the results that were obtained after protein docking protocol was run. Let me just show it to you. Yeah. So this is the protein docking table that is observed. So you have X, Y, Z coordinates here, which is the translational coordinates, translational vectors associated with a particular docking solution. You have IX and JX, which are the rotational vectors associated with that particular docking solution. You have a grid-based scoring function here, which is the score. You have contact fingerprints. So based on these contact fingerprints, there is a clustering that has been done by the software. So this is based on this contact fingerprints data. Other than that, you have electrostatic energy, you have van der Waals energy term, you have surface energy term as well, and you have the total energy term. So it is always recommended that you use total energy term for ranking your complexes. So what I'm going to do is I'm just going to double click on this particular column, and now you can see the relevant doc pose has been loaded into my workspace panel. Let me show you that. So I can just toggle this on. So this is your SARS-CoV protein, and this is your ACE2 protein. Yeah. So now what we did is we ran MD simulation for this particular pose after selection based on the energy, the lowest possible energy. So to do that, again, it is very, very simple to have this setup. You can just click on this doc pose one. You can just go to mole mechanics, click on run MD simulation. If you have a membrane behind your proteins or ligands, then you can use this particular option wherein you can add the membrane as well. But since it is just protein for me, I can use this particular option. I can use object to MD. I have to select as my doc pose. I can use this solvate option, which will add water molecules, TIP3P water molecules into the structure. I can use this relax option. This is like the minimization step, wherein it will avoid all the steric clashes. You can select the time you want to select. Like if you want to run it for 100 nanoseconds, you can select 100,000 depending upon your need. So for this study, we had selected 100 nanoseconds. You can also give the number of confirmations to maybe 2000. You can also increase it more if you want to, if you want more accuracy, but that also increases your volume of the data that is generated. So if you have enough storage capacity, you can always increase this number of confirmations to more. You can tick on this option, which will use the GPU if available. So if you have a GPU, it is always much easier to run your MD simulations. By default, it will select the GPU by itself, or you can also select your own GPU. Like if you are running calculations on one particular GPU, you can select the residual GPU that is remaining for your calculations as well. So after this, you can just click on OK and it will start running. Again, since MD simulation is a longer run procedure, what I'm going to do is I'm just going to take you to the results that were obtained. Yeah. So after MD has been completed, you will see, pardon me. Yeah. You will see this object that is loaded, doc1 pose, doc pose 1 MD. So this suggests that the MD simulation has been carried out. Let me just click on this. Yeah. Now, if I want to view the trajectory, the video, I can just click on this green button. So this will animate the stack confirmations. So this stack is essentially all the trajectory frames that are condensed together and put it into one particular object. Now, if you can see, there are some motions here. Yeah. So now if I want to if I want to calculate certain parameters, what I can just do is right click on the stack, click on stack calculations, and then I can use all these different parameters for my own selection of the stability of the complex. So if I click on RMSD to reference, what I need to do is first align the pose, align the first pose, that is my first stack confirmation, along with the docking pose. So essentially, I'm using the initial pose of that obtained after docking before MD simulation run as my reference pose. So I can just do that using this particular option. Yeah. And I can just click on superimpose. Yeah. So after I click on superimpose, it will ask me to generate RMSD. I can use this option, can just make a selection in the reference object. I can click on OK, and that will give me the RMSD graph. So here is the RMSD graph of the complex. So we have plotted RMSD for the spike protein, the ACE2 protein, as well as the complex. So if you see here in this graph, you can very well see that the spike 2 protein is fluctuating a lot more. But when it is available as a complex with ACE2 as a protein, it is showing much lesser RMSD score, or RMSD value rather. So that means that the complex in itself is very, very stable. When the spike 2 and ACE2 are interacting, it is very, very stable. Similarly, you can also calculate RMSF. So if I give a right click here, stack calculations, RMSF, I just need to select the particular residues, green selection for residues for RMSF calculation, orange selection. But if you do not make any selections here also, then also the RMSF will be calculated for your respective protein structures. So after you click on OK here, you can now see RMSF. So we have again calculated RMSF for both the spike protein and the ACE2 protein. So in the spike protein, now you see the RMSF is showing that there is a little higher level of flexibility. So this flexibility may be owing to the fact that it is trying to accommodate itself into the ACE2 binding site. If you compare it with ACE2, you can see the RMSF value is very low for about 3 angstroms, whereas for spike protein it is slightly above 3, above 4 angstrom as well at some places. The terminal residues, as usual, they are known to be flexible, and that is what is showing in these particular RMSF graphs also. The terminal residues are flexible. But overall, if you see, even if the spike protein is a little flexible, it is trying to accommodate itself into the ACE2 structure. So that is what we can see using these RMSF graphs. We also further calculated the interactions of the ligand, that is the SARS-CoV protein, with the ACE2 protein. So to do that, you can just give a right click here again, stack calculations, click on ligand contacts, and just click on OK. You can see that there are different contacts here that are formed. Apole stands for non-polar interactions, HB stands for hydrogen bonding interactions, negative are negatively charged interactions, positive is positively charged interactions. So these multiple residues, they are forming interactions at different time frames. So you can see that there are multiple different interactions that are formed. And now if you want to design certain inhibitors for this protein-protein interaction, you can always target these particular amino acid residues. So you can target those using different approaches. Like you can go ahead for protein engineering, wherein you design your own protein. You can go ahead for a small molecule inhibitor which can interact on these particular residues. So there are multiple different options for you. You can also design a peptide which can act on these residues. Yeah. Other than that, we have also calculated radius of gyration. So radius of gyration is radius of gyration is it tells you about how compact your complex is. So if you go if you know that it is showing a very stable sort of a graph across 100 nanosecond time frame, you can know that the particular protein complex is very stable. It is compact enough and it is not showing a lot of flexibility, which in itself is telling you that it is telling you that it is forming a stable complex. Other than that, you can also go ahead for contact area calculations. If you give a right click here again, stack calculations, area, you can select, you can use this green selection and orange selection. So ligand, you can select it in orange like this, and you can select this protein as green, and then you can just click on OK. So contact areas are very important parameter for protein docking, since since the residues they are involved are only on the surface. You need to have a proper idea about what are the changes in the contact areas. But if you see throughout the graph, there are very minimal changes in the contact areas, meaning that the conformations of these proteins, they are in an idealistic conformation, which are maintaining these contact surfaces throughout the time run. Yeah. Before that, just before winding up, let me come to a fact that I have missed. I wanted to compare the RMSD between my doc posts, which is here. Yeah. So I can compare my RMSD between the doc posts and the 6M0J. So let me load that molecule. So as I said, this is a crystal structure which is already loaded into the PDB database. It has both ACE2 and the spike protein. So let me just convert this into an ICM object using the same options. So now I can just superimpose both of them. So if I select 6M0J and if I select my doc pose and I can go to display, can click on superimpose and I can superimpose it on the doc pose. So I already have the result for that. Let me just show you that. Yeah. So this is the result of superimposition. The RMSD obtained was about 1.52 angstroms. This is very well within the range for protein-protein docking complex. Yeah. So that is all about the protein-protein docking complex and the ACE2 and SARS-CoV-2 study that we undertook. So if you have any questions, I'll be very happy to answer them.
Thank you so much, Nechit, for the detailed and highly informative session. We'll now move to Q&A session. We have received few interesting questions from our audience. Stick with one minute. First question is, which interaction are the most prominent in protein-protein interaction?
So the most prominent interactions among the protein-protein interactions, they are essentially these interactions, hydrophobic interactions, electrostatic interactions, they are more prominent. Other than that, you also have hydrogen bonding interactions, van der Waals interactions, and all of those as well. Yeah.
Okay. And second question is, how are the ODAs calculated?
Yeah. So ODAs are calculated as desolvation energies for those particular residues. So if you have a higher desolvation energy, that means that that particular protein, that particular residue can form interaction with the other protein residues. So it is calculated in that particular sense. Yeah.
So next question is, what is the role of calculation of contact areas in protein-protein interaction?
Pardon me, can you come again?
Is the role of calculation of contact areas in protein-protein interaction?
Yes. So contact areas, as I told, they are very, very crucial since protein-protein interactions, they only occur on the surfaces. It is important that you know what are the contact areas between those two proteins rather. So that is why contact area, they are very critical. So if you have a stable graph for your contact areas, that means that that particular protein-protein complex is a stable complex.
The last question is, what is the acceptable RMSD cutoff for protein-protein docking complex?
Yeah. So the acceptable cutoff for protein-protein complex RMSD, it is based on 2 to 3 factors. So Capri challenge that we have undertook as an ICM modes of submission, it was ranked top, and there have been certain parameters that have been stated by Capri. So if you have interface residues, if they are within the RMSD of about 2 angstrom or 5 angstrom, they are supposed to be acceptable. Or if you have a ligand, that is your protein in this case, uh, if it is having an RMSD of less than 10 angstrom, it is an acceptable sort of a range. Yeah.
Okay. Thank you so much. And thank you all for the excellent and thoughtful questions. Your active participation truly makes this session more engaging and meaningful. While we try to address as many questions as possible, a few may remain unanswered due to time limitation. Don't worry for that. Our team will review all the pending questions and get back to you via email and with the detailed response. We come to the end of today's webinar. On behalf of Advent Informatics, I extend a sincere thank you to Mr. Nachiket for sharing his expertise and guiding us through the interesting topic, molecular interaction between the spike protein and ACE2 receptor using advanced approaches. To you as well, our all participants, for your time, engagement, and active participation throughout the session. Forward welcoming you again in our upcoming webinars with more insightful scientific discussions and hands-on learning experiences. Till that, have a wonderful day ahead. So much.
Thank you so much.